It's my great pleasure to introduce Jack Johnson to you. Jack is the Eldon R. Strom Professor of Structural Virology here at Scripps. Jack originally hails from Oak Ridge, Illinois, which is near Chicago. He attended Carthage College in Wisconsin, where he obtained a bachelor's degree in chemistry, then moved on to Iowa State University to pursue graduate studies, also this time in physical chemistry, and joined the laboratory of Bob Jacobson. In his laboratory, Jack focused on crystallography and structure determination of small inorganic compounds, I think, or were they organic? Organometallic compounds. And using X-ray diffraction. After obtaining his Ph.D., he joined the research group of Michael Rossman at Purdue University in Indiana, where he took on a project that involved virus crystallography. And that's where Jack's career in virus crystallography and virus structure determination began. In fact, Jack was the first member of Michael's lab to take on a virus crystallography project, which at the time required pretty heroic effort. It was in the 70s. Jack prepared and crystallized southern bean mosaic virus and solved the structure of this virus. And within a few years, he was offered a faculty position at Purdue University and set up his own virus structure determination group, and very quickly rose to the rank of full professor. So when you read his CV, you think this was somebody who was firmly rooted in the Midwest, and he wasn't really looking for change. However, change happened, and this was a result of a sabbatical visit that Jack spent here in Ian Wilson's lab in 1993. And before Jack knew it, he had been recruited to Scripps and joined the faculty himself in 1995. So Jack's career in structural virology has been nothing but stellar. It now spans 40 years, and it's impossible for me to summarize in a few sentences all the achievements and contributions, but I thought I'd highlight a few that I witnessed during the last 25 years. Jack's group was the first to not only visualize the high-resolution structure of a protein capsid of an icosahedral virus, but also visualized significant portions of the encapsulated genome, which was a major achievement at the time and still is. It has remained an enigma, really, what the structure of encapsulated genomes in virus particles is, and Jack made a significant contribution to that. Also, he and Tim Baker, when they both were at Purdue, Tim Baker is now at UCSD, pioneered an approach that is so routinely used these days that nobody thinks about anymore, that somebody had to come up with this idea. And that involves the combination of X-ray crystallography and cryo-electron microscopy to tackle structural analyses of very large protein complexes, multi-layer protein complexes that could not be crystallized. And so Tim and Jack together used X-ray crystallography and cryo-EM to find out what the antibody binding site on a virus particle was. And today this approach is used commonly. Jack's work on bacteriophages, but also insect viruses, has illuminated virus maturation. The fact that viruses mature in the first place, that they use different pathways, and that the maturation can often involve dramatic reorganization of viral capsids. And the idea that virus particles are dynamic is really something that he championed and turned out to be very important for biological function of virus particles. And the list goes on and on. Jack's lab has solved so many virus structures that his work has uncovered relationships evolutionary relationships between viruses that were completely unanticipated, based on the fact that these viruses infect organisms from completely different kingdoms, bacteria all the way up to mammals. So I could go on and on, but I don't have the time. And suffice it to say that Jack is a leader in the world leader in structural virology, and that he has been a a figure who has made important contributions, is still making important contributions in this field. His discoveries have been published in over 300 papers, many of them in Science and Nature. He has trained 65 postdoctoral and predoctoral fellows, many of whom now hold independent faculty positions. His expertise is widely sought. He is a member of the editorial board of several scientific journals. He serves on numerous advisory boards, including the Advisory Council of the Institute of General Medical Sciences at the NIH, and is just really a very busy individual. So we're very grateful that he could take time out of his busy schedule to give this lecture tonight. And we'll talk about virus maturation and how it relates to protein dynamics.
Thank you, Annetta. Usually when I do these introductions, I just butcher the daylights out of them, and there's a number of witnesses that can say that this did not fall into that category. Thank you very much. And I should say that much that's happened in the last 16 years at Scripps has been a very, very close collaboration with Annetta, and the combination of her extraordinary molecular virology and some of the biophysics methods that we brought forward has really been a marvelous, marvelous collaboratory time. So I'm going to talk about viruses, and just to get things onto the size scale, for those not familiar with how small they are, they generally tend to be in the 100 to 200 nanometer range of size, and the techniques that we use to study them structurally are crystallography and electron microscopy. And those will be the dominant methods that I'll be describing today. So a few years ago, it really dawned on me how important virus maturation was. And so just to give a definition here, it's the process by which a virus particle transitions from a non-infectious, often fragile and spherical initial assembly product to a robust, faceted, infectious virus. The physics and chemistry are well understood for only a few systems, but in those systems, they really do reveal some novel mechanisms for creating biological dynamics and for through landscape adjustment, energy landscape adjustment, and then through Brownian ratchets. And both of those are present in the systems that we're looking at. So the thing of it is that that procapsid has encoded in it a program that brings it from that non-infectious state to the infectious state. And that program is something that we've really been interested in decoding and understanding. And just for the sake of the broader spectrum of things, virtually all animal viruses and certainly bacteriophages undergo virus maturation. And probably the one that's best known is the maturation of HIV, and the protease inhibitors stop that maturation and render the particle non-infectious. So it's these kinds of large structural changes that we're interested in, only in a system where we can really tease out a fair amount of detail. And nature being the way that it is, we can't do all of the studies that we want with one system, so we've focused on two systems. And one is a bacteriophage called P22, and it has a diameter of 650 angstroms, 65 nanometers. There's a total of 9 gene products that make up this particle, and the genome size, which is double-strand DNA, is 43,000 base pairs. This is a system that we've looked at exclusively using electron microscopy. The other system that we've looked at is a lambda-like bacteriophage called HK97. And if we just look at the head, they're actually very similar to each other. But they have totally different tail structures. And in this system, we're not going to be worrying about the tail structure. We're going to be worrying about how we can follow the dynamics of virus maturation. It has a very similar size in terms of its genome and diameter as P22, but in the system that we look at, there's only going to be 2 gene products initially and then 1 later. So we're going to start with P22. And one of the features of this bacteriophage is an enzyme that can cut its way through the outer peptidoglycan that covers the Salmonella host that it normally infects. It brings it down into juxtaposition with the host cell surface, where it then delivers the DNA. And in about 24 hours, you have a burst size of virus particles in the Salmonella on the order of a few hundred particles per Salmonella cell. So these viruses replicate at exceptional levels of efficiency. Now, viruses, by their nature, having a limited amount of genetic information, have to have great genetic economy. And this is accomplished by using many copies of a few gene products. And this is a cartoon that we'll see in a little while is actually quite accurate as to what the processes are associated with the assembly of bacteriophage P22. Initially, there's three gene products that are really important to the capsid. There's this dodecameric portal protein, which is gene product 1. We have the capsid protein, which is gene product 5. And we have a scaffolding protein that's gene product 8. And really, very little is known about how this initial association takes place. But once it takes place, we end up with our pro-capsid that has the scaffolding protein on the inside, the capsid protein on the outside. There's a few other gene products that are resident already at this point in time, but there's no DNA inside of this particle. And in fact, what happens is there's two other gene products, GP3 and GP2, that attach to the portal and then pump DNA into this preformed particle. During that process, the scaffolding protein is released and actually recycled into the next round of particle formation. And then the DNA is inside at a very high pressure, about 10 times the pressure of a champagne bottle. And that has to be held inside of this particle while these closure proteins, GP4, GP10, and finally GP26, are attached to the portal to give you your final infectious particle. So areas that we're interested in are these transitions here, and then we've been very interested in the final mature particle of P22. So one of the techniques that we utilize a lot is cryo-electron microscopy, and we are incredibly fortunate at Scripps to have one of the truly outstanding facilities run by Bridget Carragher and Quint Potter. And this is the sort of image that one gets of the procapsid of P22 if you image it in its frozen, hydrated state at about 60,000 magnification. Now, there's two gene products in this particle. GP5, the capsid protein. There's also scaffolding in here, which I didn't indicate here, and the portal. Now, these particles all have icosahedral symmetry. So we can ignore the fact that there's a portal in these particles, and we can average over the icosahedral symmetry in the individual particles, and we can average all of these different particles to generate a much higher signal-to-noise ratio than what we see in any given individual particle. And when we do that, we end up with an image, in this case at about 15 angstroms resolution, that clearly gives us definition of each of the viral subunits. And it also shows us that these subunits organize in hexamers and pentamers, and the overall organization of this particle has what we call T equals 7 symmetry. There's a total of 420 subunits if you ignore the fact that one of the pentamers is actually replaced by the portal. So indeed, one of these pentamers is replaced by a portal, but because you have 11 other pentamers that average together, the portal just sort of disappears in this averaging process, and we end up with something that looks truly icosahedral. Now, one of the beautiful things about cryo-electron microscopy is that the density that we generate is exactly like X-ray crystallographic density, which, unlike negative stain, where you only see the surface, with the cryo-EM, we can cut through and look on the inside of the procapsid, and indeed, it's empty. And in red, we're seeing some remnants of the scaffolding protein that was required to allow this initial assembly to take place. I should point out, without that scaffolding protein, you end up with particles that are highly deformed and very irregular in shape. Now, one of the experiments that we did was to align as best we could the positions of these portals. And when we did that, we were able to come up with a so-called asymmetric reconstruction of the procapsid. And this just illustrates the fact that we have these other two enzymes that bind to that and then pump the DNA into this particle. So now if we look at a fully mature form of P22, we see particles that look like this in our cryo-EM image. And while they're not visible in these particular particles, these have that full tail machine that I had described in the cartoon. But again, we can ignore those tail machines, and we can just average with icosahedral symmetry. And when we do that, we get a reconstruction of the fully mature particle with no visible tail machine because of the fact that we essentially averaged it away. Now, when we cut inside, we can clearly see the layered double-stranded DNA that is packed at liquid crystalline density inside of these particles. And if we compare the structure of the procapsid on the outside that I showed you earlier with the mature particle, and these are on the same scale, we see that these particles go from about 500 angstroms in diameter to 650 angstroms in diameter, and all of these openings where the scaffolding protein leaves the particle actually close and becomes a very tightly sealed particle in the fully mature form. So you get a sense of what the dynamics are like when this particle goes through that transition. Now, a number of years ago, Gabe Lander, as part of his Ph.D. thesis, determined the 17 angstrom structure, 17 angstrom resolution structure of the asymmetric P22. And this involved the about 26,000 particle images, nearly 5,000 micrographs, and an enormous amount of individual effort in computing. But this was all done using the automated microscopy facility that we have available here. And in 2005, I'm not sure we could have collected this amount of data without that type of facility. But this now allows us to go inside and look at what the particle is like on the inside. And we can control the contour level. So we can raise the contour level to get rid of everything that's disordered. And now we can clearly see individual rings of double-stranded DNA as it's packed against the capsid surface. We get a beautiful view of the dodecameric portal, and we can see actual density that is located in the portal structure itself. Now, when we solved this structure in 2006, we completely misinterpreted what this density is. And we thought it was something called pilot proteins, but as we'll see in a few moments, this density is now fully interpreted in its proper way. This movie gives us a kind of a summary of the information that we have in this asymmetric reconstruction. These are the enzymes, the GP9 enzymes that are required to get through that peptidoglycan. These are adapter proteins that bind to the portal. That's the closure protein that keeps the DNA inside. We see this really strong ring of double-stranded DNA right around the portal structure. And as we cut through this density, we can see the individual layers of DNA as they exist inside the particle. This is the DNA delivery, both the packaging and delivery machine. It sits in this pentavalent site on the capsid of the protein. Itself, it has 12-fold symmetry, and then it adds these additional proteins to it. So this was circa 2006, and it allowed us to identify all of the different gene products that I had shown you in the cartoon, with the exception of that region that we misinterpreted. Now, there's a lesson here. One of the things that they do in NRAM is they archive every image that's ever collected. So in 2006, we did the best that we could, but between 2006 and 2010, there were many, many publications improving the clustering and so forth of the particles, and using the identical data set that we used in 2006 to get 17 angstroms resolution, we got 7.8 angstroms resolution in 2010. And this allowed us to get considerably more detail. We could count the rings of the DNA as it existed in the particle. But much more importantly, a former postdoc in the lab, Gino Ciungolani, was busy solving the structure of that portal protein as an expressed, isolated gene product. And when Gino solved that structure, we were all shocked to see that in addition to the polypeptides that form the ring, the dodecameric ring, the C-terminal portion of that sequence formed this amazing 12-fold coil that extended 200 angstroms above the surface of the protein, of the normal portal. And what's more, he also truncated that and got a higher resolution structure of one of the adapter proteins bound to the portal structure. So Gino's crystallography, combined with the fact that we got this higher resolution structure, was really exciting, because now we could take that X-ray model and we could put it into our higher resolution cryo-EM model. But there were some problems, because it didn't fit properly in this region of the structure. And also, if we looked at our structure where we had helices, everything was exactly out of phase if we took that X-ray model and put it in. However, we're structural biologists, so we make a few adjustments, we get it to fit. And when we did that, we were able to come up with a very nice fit, and all of our helices now were in register with the helical density in the cryo-EM reconstruction. And comparing the two structures became very interesting, because in the fully extended state here, that's Gino's structure. And this compressed state is our structure. And there's no real inconsistency here, because our structure is in the presence of liquid crystalline DNA, and the Gino structure was as an isolated protein in solution. So what we're looking at, I believe, and we convinced the referees, that we're looking at the two conformations that exist for this protein depending on what its environment is. And most interesting is the fact that in this compressed state, it actually closes down that opening for where the DNA comes out. And in fact, in the higher resolution structure, we can see DNA all the way down this, down the barrel, as we call it, and into the portal region of the structure. So this is circa 2011, and as I said, that we had wrong before, that we now know is actually part of the portal itself. But it's interesting, because this particle has been studied for 50 years, and it was only about 2 years ago that we realized that, in fact, we had this entire extension onto the portal, and that it probably has a really important functional role. So back in 2006, when Gabe Lander was solving the structure as a graduate student, and I mentioned before, and I emphasized that Bridget Carragher and Clint Potter are absolutely essential for allowing us to get this structure. Sherwood Kazins provided us with all of the particles that we used. Peter Privilege provided the portal proteins and a number of very insightful ideas as we were going along. Liang Tang actually got the first asymmetric reconstruction of P22 just as he was leaving for a faculty position at the University of Kansas, and then Gabe took that and went to higher resolution. So this is now, in the last couple of years, Gino Cingolani at Thomas Jefferson University did this crystallography, and then Jinghua Tang and Tim Baker's group at UCSD extended the resolution from 17 to 7.8 angstroms. And this is just Gabe's interpretation of the DNA flowing into the particle through the barrel of the portal. So most of what I was telling you about were static structures. And now what I'd like to do is to bring these structures to life. And that's what HK97 allows us to do. And I will explain why in a moment. So HK97, as I said, it's a lambda-like phage. It's a feeding frenzy when these things are exposed to E. coli. And they are very, very prolific in E. coli. Now, I had, for many, many years, wanted to do crystallography on a bacteriophage. But that guy right there was a big problem, because you're not going to crystallize something that's got a almost 3,000 angstrom long tail associated with it. So way back in the mid-90s, Roger Hendricks and Bob Duda at the University of Pittsburgh came up with a way in which you could express only 2 genes from HK97 in E. coli, which is the natural host for HK97, and those 2 genes could be pushed through the maturation process to make empty capsids that were identical in size to the capsid that we saw in the bacteriophage. Bill Wyckoff, who was a graduate student in my group, was able to grow crystals of this capsid, 650 angstroms in diameter, and was able to determine a 3.5 angstrom X-ray map using these particles. Now, I don't think we're limited in any way anymore in terms of unit cell sizes. This was a fairly recent picture that we took of crystals of an intermediate in HK97 maturation that were over 1,000 angstroms in their unit cell parameters. This is an image taken from the advanced photon source, the GMCA beamline that has been operating there. And when we zoom in, we can see a spectacular order-to-order resolution of these spots for 1,000 angstrom unit cell. And Vijay Reddy has pushed, and Glenn Nemerow have pushed this even further with adenovirus, where they have even larger unit cells. So if you can get it to crystallize, you can collect data at the GMCA beamline. And we actually collected, this data was collected in the late 90s on the fully mature capsid of HK97. And this was done the old-fashioned way, when we didn't freeze crystals, and we collected almost 500. We used almost 500 crystals. We measured almost 22 million reflections to give us a data set that was about 5 million reflections. And we never made any heavy atom derivatives or anything. We used the cryo-EM reconstruction as the starting model, and then we used the symmetry of the particle, the icosahedral symmetry, to do phase extension. So we phase extended all the way from about 20 angstroms resolution to 3.4 angstroms resolution. And when we did that, we got really beautiful electron density. And one of the interesting features of this is that we have so-called quasi-equivalence. So we've got the same gene product that forms hexamers and pentamers, and we can, these are icosahedrally independent from each other. So you can make comparisons. You've got an internal control to see whether you have done the experiment properly. But there's 7 subunits here, 1 from the pentamer and the 6 that are in this hexamer that are independent as far as the icosahedral symmetry is concerned. And when we superimposed those subunits on top of each other, you can see that the tertiary structures of these were virtually identical with just a little bit of motion. This is important because we're going to come back and we're going to look at this again with a different assembly product where this is not the case. The fold of this subunit at the time was unique. It hadn't been observed before. It has an alpha-beta structure in the region that forms the hexamers and pentamers. It has this beautifully characteristic 40 angstrom long helix with a loop out of that helix between 2 of the turns. I remember very well Peter Wright coming and looking at this model and said, something is very wrong here. Helices don't do this. I think I'll be able to explain why they do it today. But in fact, they all did it. All 7 of them did it. So Peter's worries notwithstanding, we move forward. Anyway, you'll notice that the first residue here is amino acid 104. There's a good reason for that, and we'll see it in a few moments. One of the other novel features of this structure was that in the capsid, it undergoes this autocatalytic ligation to form an isopeptide bond with the loss of ammonia. And the 2 residues that are involved are this asparagine and this lysine. And they're critically important. There's a 3rd residue, this glutamic acid, that is essential for the reaction to occur. It is autocatalytic. No enzymes involved in this whatsoever. Now, at the time we solved this in 2001, it was the only structure that had that fold. Since then, many different subunit folds have been determined from double-stranded DNA bacteriophage, from some assembly products in bacteria that form these containers for enzymes. And in fact, the HK97 fold, as it's called, has shown up in all of these. So this is sort of the canonical fold for double-stranded DNA viruses that we see that go through the bacterial viruses. And I guess we should have expected it since it happened that way with other systems, but we were still shocked when this fold has shown up over and over and over again. Now, in the fully mature particle, each one of these subunits is actually layered on top of its neighbors. And this was a real problem when it came to tracing the polypeptide chain at 3.4 angstroms resolution, because you could easily jump from one subunit into another. And in fact, it wasn't like any organization of protein that we'd ever seen in a virus before. And so it immediately suggested why there needed to be intermediates in this process, because it was impossible to envision how you could get proteins to go together in this way in one step. So that really suggested what the maturation was doing for us. The other thing that was peculiar about this particle was that it had the shell was only 18 angstroms thick. And the particle at its maximum dimension was 660 angstroms. So it's kind of like a molecular balloon, a very thin shell with a very large volume inside. And this is where the crosslinks that I mentioned come into play. Because these crosslinks aren't just any old types of crosslinks. These crosslinks actually form a fabric of interlocking hexameric and pentameric crosslinks. So all of the subunits that are in the same color here are cross-linked together to form what we call the superhexamer and the superpentamer and so forth. And this matrix cannot be disassembled without treating it with a protease. So it gives this particle enormous stability and, in fact, eliminates the need for some of the accessory proteins that are seen on other bacteriophage. Essentially, gene products have been replaced by chemistry in this particular situation. Okay, so everything that I've told you about has been what we call head 2, the fully mature particle. Roger Hendricks and Bob Duda had actually mapped out the existence of a series of intermediates. And the two genes that are expressed in these virus-like particles is a virally encoded protease and the capsid protein. The capsid protein spontaneously forms hexamers and pentamers, and they assemble and they package the protease inside the particle. Now, if that protease is active, you never see this particle, because as soon as the particle forms, a proteolysis initiates, and it cuts off the subunits at residue 104. It deletes the entire N-terminal 100-plus amino acids. However, we can make mutants of the protease that are not active, and we can purify this particle, and we have a 5.2 angstrom structure done by crystallography of that. So this is what we call PROHEAD 1. It's 19 megadaltons in size. After the proteolysis, we lose 6 megadaltons because we lose 420 times the equivalent of 103 amino acids, plus the protease auto-digests. And all of these fragments diffuse out of the particle. So the N-terminal region diffuses out, and the protease fragments diffuse out. And in our expression system, this is what we purify if we have an active protease, this so-called PROHEAD 2 particle. Now, in vitro, in vivo, this is the substrate for DNA packaging. So the enzymes are going to pump DNA into this particle. And when that happens, it goes down through this maturation pathway. In vitro, we can recapitulate this by just lowering the pH to 4. And then we can drive that particle right on through all of these maturation intermediates. And we've been able to do structures of all of these different maturation intermediates. So PROHEAD 1, we've got a 5.2 angstrom structure. PROHEAD 2, we have a 3.8 angstrom structure. Expansion intermediate 1, 8.5. What we call a balloon, which is almost there, 3.8. And then the structure that I described to you already at 3.4 angstroms. So now I'd like to focus on this dynamic part of this process. So this is a cryo-EM reconstruction of PROHEAD 2. So this is now after the proteolysis, but before the DNA has been pumped in these particles. If we look at hexamers on this subunit, on this particle, they're not really hexameric at all. They're sort of like two trapezoids related by, and each one has 3 subunits in it, related by a 2-fold axis. And after maturation, these become a beautiful hexameric structure. Now, we took that cryo-EM reconstruction, and if you look at the sort of outline of the capsid, you can see it's very corrugated. And we took our high-resolution model of the subunits, and we modeled it into that procapsid as best as we could. As you'll see in a few moments, it wasn't a very good job. But it gave us some ideas that we could use to design experiments. But this is what the procapsid profile looks like, inside, outside. And that's what the mature capsid looks like. So it's like you've taken the procapsid corrugated structure and you've ironed it out. And this is extremely uniform, doesn't have any of that corrugation in it. And I can tell you, just as a heads up here where we're going, the procapsid structure assembles subunits like this. The subunits come together like a palm-to-palm interaction in my hand. And in fact, the envelopes of those subunits are very nicely defined, very unlike what we see in the mature particle. And what maturation does is that. And that expands the particle, and it becomes thin. So it's a relatively rigid body, but not entirely rigid body motion of these subunits that take you from 450 to 650 angstroms in diameter, and a shell that's about 40 angstroms in diameter to a shell that's about 18 angstroms in diameter. And we'll see that in a little more detail in a moment. So one of the things that building that model let us do was to make a movie of what the actual change was like that these subunits were undergoing. Now, this movie is not a very accurate movie. It's just morphing two states. None of the intermediate states are represented here. And it doesn't run backwards and forwards. It only goes one direction. So the movie is wrong in that regard. But it gives us some. So here's our asymmetric hexamers. And then if we just morph things in, we see our symmetric hexamers here. But one of the most interesting things that this movie did for us was to give us the idea of using crosslinks as a metric for where maturation was going. The lysine is here, the asparagine is there. In the procapsid, according to our model, they start out 40 angstroms apart. And when the maturation takes place, they come into juxtaposition with each other. And the third residue is coming from a third subunit that makes this cross-linking organization. So the formation of crosslinks is actually a metric of what the structure of the particle is. And Lou Gann, a former graduate student here, actually did a spectacular job of mapping out the motions of these particles by just following the crosslinks associated with it. Now, I told you this kind of hands-folding process. Well, this is a Hoberman sphere. And the Hoberman sphere is made up of these struts. And if you look at the low radius form of the Hoberman sphere, these struts are more or less radial. And if you look at the large diameter, the struts are more or less tangential. That's exactly the motions that these subunits are going through as you go through this transition. So this is actually a pretty decent mechanical model for what is going on. Now, I want to take the last few minutes here to actually look at some of the kinetics and dynamics of these transitions. And what we want to focus on is this transition from PROHEAD 2 to expansion intermediate 1. And Kelly Lee, who was in the lab a number of years ago, thought, given the fact that these things change size, small angle X-ray scattering would be a very nice way to look at this. And in a very simple explanation, if you have a large sphere, you have a compact sort of target image scattering. And if you have a small sphere, it expands. So if you start with a small sphere, in other words, PROHEAD 2, and you allow it to expand, what we expect to see is the diffraction pattern is going to get more compact. And what Kelly did then was to add a third dimension to the experiment, and that was time. So using a stop flow apparatus in collaboration with Hiro Saruta at SSRL, we were able to record Sachs patterns at 7-second intervals after we initiated the expansion by lowering the pH to 4. And this was done in a very precise way. And this is the pattern that we had from our procapsid. And at 7-second intervals, we can see each of these intermediates, which I thought were intermediates. The biophysicists in the crowd will realize that these are isospestic points. And what isospestic points tell you is that it's a two-state system. And that we are not looking at intermediates. What we are looking at here is a stochastic process where these particles are going from one state to the other state. And these are linear combinations of the diffraction from those two states. And I wasn't clear on this. Kelly spent a lot of time explaining this to me. And finally he said, and this was a good example, he said, let's think of popcorn. And I said, okay. And he said, if you want to look at popcorn popping, you're not going to learn much by putting it in the oil in a kettle and turning on the heat and watching the kettle fill up with popcorn. He said, all you're going to learn is how fast is the popcorn popping. And he said, this is a kettle experiment. We're watching how fast that kettle is filling up with mature or expansion intermediate 1 particles. If we want to look at popcorn popping, we've got to get an isolated popcorn, and we've got to get a very high-speed camera to look at that. Because no matter how you do this experiment, you're never going to be able to detect intermediates the way it's set up. It also had a very important other component to it. It implied that this PROHEAD 2 particle was actually metastable, and that by lowering the pH, it spontaneously jumped over an energy barrier and went to this expansion intermediate 1 particle. So it immediately suggested to Kelly that PROHEAD 2 was metastable. Now, we recently got an 8.5 angstrom structure of the expansion intermediate 1, that particle that is the result of this transition. And in fact, we see that the subunits are already now in perfect hexameric form. The skewed hexamers have symmetrized, but this particle still has a way to go. But we're not going to worry about that for the moment. What Kelly did was to go back and say, OK, let's watch popcorn pop 1 kernel at a time. So he came up with a very ingenious way of layering polyethylene glycol and a very dilute amount of biotin-labeled polyethylene glycol on a glass slide. This was a very, very hard experiment because every time he took PROHEAD 2 and tried to attach something to it, it popped. These were things waiting to pop. And in fact, that comes back again in just a few moments. But with a lot of effort, he was able to attach PEG about 1 copy per particle, and then added the particles and streptavidin and immobilized these particles. And then we could look at individual virus particles. These were just labeled with Psi5 fluorescent dye. But we could clearly see the individual particles undergoing their thermal motion on the surface of that glass slide. Next experiment was to find a readout of the state of that particle. And we invested a lot of money with molecular probes, but eventually Kelly found one that showed almost, this is a bulk experiment, showed almost no fluorescence for the procapsid. And then when he lowered the pH, the fluorescence rose dramatically. And then that allowed us to do the following experiment, and I hope you can see it. We can now watch popcorn popping 1 kernel at a time. This thing just keeps recycling. I hope people, at least near the front, can see that these particles are appearing, but it's not real important because what we can do is we can go back and we can interrogate the pixels, and now we can watch individual particles going through it. And in fact, it was exactly as Kelly had described from the Sachs experiment. It's a stochastic process. You lower the pH. Some particles undergo the transition within a few seconds. Others sit around for 3 minutes. And the actual transition that it takes for each particle is about 1 second, even though the half-life is about 2.5 minutes under the conditions that the Sachs experiment, actually the half-life is about 1.5 minutes under the conditions that the Sachs experiment was done. So now this allows us to actually watch popcorn pop 1 kernel at a time, and again, it re-emphasizes this. Now, Kelly found a paper. It's amazing how once you get an idea in your head, somebody's already demonstrated it. So back in 1993, Jonathan King's lab at MIT was looking at the maturation of P22 using calorimetry. And what they showed in this Biophysical journal paper was that, in fact, it was exothermic. And when that particle went through its expansion, it actually gave off about 21.5 kcals per mole of capsid protein during the expansion. Now, we haven't been able to do that experiment with HK97, but I am a person of faith, and I think it's there. So the question then becomes, where does the energy come from? How do you set up an energy landscape so that this maturation is a downhill process. And that's where the crystal structure of PROHEAD 2 came in. Because we had built this model based on our mature particle, but we had no idea what the details of this were. And Ilya Gertzman was a UCSD graduate student in the lab, and he initially grew crystals of PROHEAD 2 that diffracted to 5 angstroms resolution. We had huge problems with these things maturing during the attempt to crystallize it. And one of the breakthroughs for us was that Bob Duda and Roger Hendricks were able to screen for mutants that were, in their words, reluctant to mature. So we were able to get a mutation that was considerably more stable in the PROHEAD 2 state than in the normal structure, or in the normal sequence. And it was actually that we got the 5 angstrom diffraction. And then Ilya went through and did some pretty serious surgery on the surface of the particle, because from that 5 angstrom structure we knew where all of the interfaces were, and he managed to reduce the dynamic character of those interfaces by deleting portions of the capsid protein. And in the end, he got a crystal that diffracted to 3.65 angstroms resolution. Now, the structure of that was really quite astonishing for us. So this was one. There's 6 subunits. There's 7 subunits. This is just one of the subunits. But when you look at this subunit, you immediately, if you've looked at these as long as we have, realize that there's something really wrong with that helix. If you didn't like our original helix, Peter, you would hate this helix because it's bent and it's twisted, and there's just all kinds of stuff that's not quite right. And PROHEAD 2, again, the subunits are organized so that they're more or less radial, and in HEAD 2 they have become more or less tangential. So this fit very nicely the model that I described to you previously. But now we had really high resolution information on our PROHEAD, and we could look at some of the details. Now this is the PROHEAD structure, and so this is one of the trapezoids there and the other trapezoid there, and now we can actually see what happens to these subunits when it goes through this transition. So there's an enormous change in its orientation. It's going out, but as we'll see in just a moment, there's a huge change in the subunit interfaces between the procapsid and the mature capsid. And now, if we zoom in at one of these subunit interfaces in the procapsid, we can see there's an extensive amount of interaction between residues on this helix and residues on this strand and this turn up here. And when it goes through the maturation, all of these are broken. And so now when we go and expand it out, you'll see that all of those interactions are broken during that transition. And just what the thermodynamics of all this is at the molecular scale we're still dealing with. But essentially what's happened is that the subunit, when it goes from the PROHEAD organization to the head, is it twists about 35 degrees around 3 strands, and then the helix has to accommodate that, and that happens by bending. So this is just one subunit first in the PROHEAD 2 state and then in the mature state. And these are all of the tertiary structure changes that are taking place during that transition. So while the PROHEAD 2 structure is clearly recognizable as the HK97 fold, comparing it to the other form is it's dramatically different. And so then what we believe is that in this distorted sort of structure that we see, and it's not really distorted. There's nothing wrong with the phi-psi angles and things like that. I mean, it's not violating any principles. It's just in a really unusual tertiary structure. So what we think is happening here is that we have, if you consider an isolated subunit, which is a hypothetical because as soon as they get expressed, they form hexamers and pentamers. But if you consider an isolated subunit, it's got this N-terminal portion that gets cut off. And what we believe is that when those N-termini, when they interact with each other, they actually distort the subunit in the way that I just described. And in fact, this PROHEAD particle, you can make it without protease. You can take that thing apart and put it back together again. We know from hydrogen deuterium exchange that things are distorted already in this PROHEAD structure. But now the protease comes along, and it cuts off all of these regions that have forced these interactions in an equilibrium sort of manner. And now we believe that the quaternary structure stabilizes this unstable tertiary structure, and then when the DNA gets pumped into this thing, it's a downhill ride to form this expansion intermediate 1 particle. So everything that I've talked to you about is this transition right here. I'm going to take the last couple minutes to tell you that the rest of this process is actually driven by a Brownian ratchet. And it's driven by the formation of these crosslinks. And when the crosslinks, what the crosslinks are doing is they're adding directionality to a dynamic Brownian motion. And as these crosslinks form, and we have a whole lot of data on that, it's actually moving that structure down the pathway towards the larger state. So this is where we spent most of our time looking at the dynamics, this transition. So this is our PROHEAD 2 structure. To get to that structure, we go through this transition. Now, to go from expansion intermediate 1 to what we call the balloon, this is where the Brownian ratchet kicks in, and that drives the structure into this organization. And then to go from the balloon to the final mature particle is just a rather subtle transition that gets everybody into their final organization. So this, given 2 gene products, you've created a rather complex set of dynamic and energy landscape behaviors that has given us no lack of enjoyment. So this has been an effort of a number of people over a long period of time. Roger Hendricks and Bob Duda were the people that came up with the molecular biology that allowed this whole thing to get started. Alistair Stephen and James Conway did all of the early cryo-electron microscopy. And then in my group, Lou Gann teased out an awful lot of the Brownian ratchet stuff that I talked about in the last 2 minutes. Kelly Lee did all of the dynamics going from PROHEAD 2 to expansion intermediate 1. And I think Ilya Gertzman is probably a real hero here in getting a structure of the PROHEAD 2 at sufficient resolution that we were able to actually understand at the structural level what these transitions meant. So this is now 15 years, I think, into our efforts. And the grant was just renewed about a year ago for 5 more years. So anyway, maybe we'll find something new. Thanks so much for your attention, and I hope you enjoyed the story.